Good evening and welcome, everyone. We're really honored that you chose to join us for this inaugural event this evening. I'm David Dodick. I'm the board chair of the American Brain Foundation and an emeritus professor of neurology at the Mayo Clinic. For 30 years, the American Brain Foundation has invested in research across the entire spectrum of brain diseases because we believe that uncovering the common mechanism or mechanisms that underlie many brain diseases, the threads that tie them together, if you will mean that a cure for one will result in a cure for many. And this is our guiding principle. So because of this focus, we've established the 2022 Cure One, Cure Many Award. This is a $3 million multi-year research award that was designed to discover an accurate method of diagnosing Lewy body dementia. The award will provide really critical support in the search for a biomarker, or otherwise known as a diagnostic test, for Lewy body dementia, which is the second most common form of dementia. Being able to make a diagnosis during life will undoubtedly lead to novel targets for new treatments and the ability to have new treatments that will make an impact on the course of this disease. In addition, because of the interconnectedness of some of these diseases, undoubtedly the discoveries in Lewy body dementia research will inevitably inform and advance research in Parkinson's disease, Alzheimer's disease, and other dementias. And this is why we've partnered and have had the privilege and honor to partner with other leading organizations in those spaces, like the Alzheimer's Association and the Michael J. Fox Foundation for Parkinson's Research, and our research partner, the American Academy of Neurology. We've come together to advance our understanding of these diseases and with the common aim to improve the lives of people and families who are directly affected. So with that, I'd like to now turn it over to Dr. Orly Abitzer. Dr. Abitzer is the president of the American Academy of Neurology, and she's joined us this evening to say a few words. Dr. Abitzer, thank you for joining us. And please, the floor is yours.
Thank you so much, Dr. Dodick. Lewy body dementia is devastating to the people it affects and their families. And this award to find a biomarker for LBD offers the hope that it will lead to an accurate method of diagnosis and give patients and their loved ones a better understanding of their prognosis. When the AAN founded the American Brain Foundation in 1992, its vision was to invest in research to defeat brain disease and disorders exactly like this one. And today, we are one step closer to doing just that. Thank you very much.
Thank you, Dr. Abitzer, and thanks again for being here tonight. It's important. Now I'd like to introduce Susan Schneider-Williams. Susan is a former board member of the American Brain Foundation, and she was the vice chair of the American Brain Foundation. And her commitment and passion and dedication over the last 6 years to honor her late husband, actor and comedian Robin Williams, and her commitment to the American Brain Foundation is really what has inspired this entire initiative. So please join me in welcoming Susan to share a little more about the heart behind the Cure One, Cure Many Award. Susan.
Thank you so much, David. And thank you to everyone for being here tonight or today, wherever you are, and for your commitment to a future without brain disease. I'm just going to take a few minutes and tell you about how I was introduced to the ABF and a few other details after that. So in 2015, I had started talking about our experience with Lewy body dementia, and specifically I'd done an ABC interview, and our executive director, Jane Ransom at the American Brain Foundation, had found out about some of, just about me stepping out and talking about this experience. And in 2016, early 2016, the ABF approached me and honored me with their Commitment to Cures Award. And that was the beginning of me meeting this organization and finding out about their mission and vision and their approach to brain disease. So fast forward 6 years later, I've just now term limited off the board. But during these last 6 years, this project, which we are celebrating tonight, is really one of the foundational projects that was near and dear to my heart. And so specifically why is because around our experience with Lewy body dementia, the main idea or the main thread behind our experience with Robin and I is that we didn't have a diagnosis until autopsy. And what I found out after autopsy, got the diagnosis of what he and I had been searching for about a year intensely, I found out that we were not alone in that. And I was shocked that there is no diagnosis while someone is alive. And so with that experience of having felt like what this disease was like and spending the last year of my husband's life with him chasing down answers, I wanted our experience to make a difference. I wanted what he went through to make a difference for somebody. And there are about 1.5 million people out there who are affected in the United States by LBD, and that's a lot. And anyway, there could be, that's probably a conservative number. So because of our experience, I thought, well, gee, if we'd had a diagnosis while Robin was alive, the most obvious things that would be different is that he wouldn't have had to be so afraid. That's number one. Number two, we wouldn't have had to waste so much time chasing down answers. It's human nature to want to get to the bottom of what's going on. I don't think there's anyone out there with this disease who is just going to accept what it is without knowing what it is. And so that could have entered our playbook. Plus, if Robin had wanted to and opted to, he could have participated in clinical trials. He could have decided about going public with this to raise awareness. Maybe not, but at least he would have had the choice. And he could have donated to research if he wanted to as well. So those are some pretty big differences, just to name a few of what could have happened if we'd had a diagnosis while he was alive. So this award is really, it's the outgrowth of we created the Lewy Body Dementia Fund at the American Brain Foundation, and this is the very first real science award connected to that fund. And we're super excited about it because it absolutely targets going after a biomarker for LBD and hopefully an early biomarker as well. And a few things I want to outline about this that make it really special and exciting is that it's not just our target goal, obviously, and there's, as Dr. Ross and his investigators have outlined in their research program, there are many goals along the way. It's not just the science itself, it is the collaboration behind it. So it's the representatives of the organizations you see tonight here with us. It's the Michael J. Fox Foundation, Alzheimer's Association, Mary Groff Foundation, and other smaller foundations and individuals as well who've given to this. It's that collaboration behind it, understanding that Alzheimer's, Parkinson's, and Lewy body are all so intimately connected. They're connected in ways that what could happen with this award and the science that the investigators will be doing is they could come up with something that has a very big effect or even a small effect, but important on Alzheimer's and Parkinson's, as well as Lewy bodies. So the whole space together. So we really see this is our inaugural Cure One, Cure Many Award, and we're just all really excited about it. It's been everyone on here tonight and many who are not, that you can't see visibly, but have worked on this, have worked so hard over the last several years. And I just want to give my personal thanks to all of you. You know who you are. And it's a real honor to have been a part of this. And I'm looking forward to what happens with the science. Thank you so much.
Susan, thank you so much. I mean, over the last 6 years, it's really been a pleasure, an honor, and an inspiration to watch you work. And what people don't perhaps know is that you've traversed this country more times than I'm sure you can count, and pounded the pavement to make this day possible. And so, thank you for everything that you've done to make this day possible and for what you've done for the foundation. As you shared, so many individuals with Lewy body dementia and their caregivers endure needless suffering because of a delay in diagnosis and indeed oftentimes a misdiagnosis. And so currently, as you know now, the only way that Lewy body dementia is definitively diagnosed is through an autopsy. And without the diagnosis, a definitive diagnosis, family lose that valuable time, as Susan just said, with their loved one, not to mention the confusion and the frustration that comes of having a disease that's an unknown disease, not really being sure what's happening, and the potential actually for complications that might arise from treatments that are given because of an incorrect diagnosis. So for many, many reasons, we need a biomarker. So we spoke to people impacted by Lewy body dementia, and Tima Berkey is here tonight to share about her family's experience. So Tima, thank you so much for being here. The floor is yours.
Thank you, Dr. Dodick. Thank you, Susan. Thank you, everyone, for having me here tonight. Thank you, everyone, for having me here tonight again. Good evening. Discovering a biomarker for Lewy body dementia is the key to early diagnosis. Early diagnosis leads to earlier treatment options. Earlier treatment options provide patients with a better quality of life. Earlier treatment options provide caregivers with invaluable time to prepare emotionally and financially for a very difficult trajectory. With continued research efforts, we will be able to identify known risk factors and treatments will improve. One day, hopefully, Lewy body dementia will no longer be the threat that it is. My experience with Lewy body dementia, as shared with my younger brother and my mother, began with a subtle clue and ended 5 years later in hospice care within a secure dementia unit. In the fall of 2008, my father detected a slight but persistent tremor in his right hand during a routine procedure. For a 40-plus year veteran cardiovascular and thoracic surgeon, this meant an early and abrupt ending to a successful and very satisfying career. The model of physical health and fitness, my dad dove full time into grandparenting and yard work. While this was initially a welcome change, it quickly became obvious that he was struggling emotionally to adjust. There were doctor's appointments and tests, all inconclusive. My parents' retirement travel plans kept getting pushed off. I'll never know the private conversations he had with my mom during that first year. I do recall that my mother, herself an MD pathologist, also felt confused and incredibly overwhelmed by all of the possibilities. Friends and extended family were noticing the subtle changes in my dad, the lost items, the spilled drinks, the non sequiturs. Was he depressed? Yes, but that did not explain the erratic behavior. Were the night terrors he had over the last few years at all relevant? Yes, but we learned that later. Was this Parkinson's disease? Not exactly. Was it Alzheimer's disease? Not exactly. We were talking in circles and growing increasingly concerned every single day. It was almost 2 years later before we had a formal understanding and a working definition of the disease that was rapidly unraveling our lives. By that point, my father was now presenting with hallucinations and irrational thoughts. Dad was getting lost on his daily walks in our tiny neighborhood where he had lived for over 30 years. The first person to mention the words Lewy body to me was my college roommate, Stephanie, a neuropsychologist at Columbia University. After I described my dad's behavior, she carefully introduced the topic. She warned me of the risk of prescribing certain medications, specifically Haldol, for Lewy body dementia. She insisted that we mention it to his neurologist. After we spoke, I did a quick search on my laptop. What I read was both an accurate description of my father's symptoms and a confirmation of my worst fears. In October of 2010, the neurology department at Penn agreed and handed us a diagnosis, which we already knew. I clearly remember later that fall day, Dad looked at me with tears running down his face, and he asked me, "Why is this happening to me?" Sadly, it was our last lucid conversation. The next day, my father started prescription medication Seroquel. The medication helped control the frequency and intensity of his hallucinations and delusional thoughts. The hard trade, which I didn't realize until after it was too late, is that once medicated, the light in his eyes never returned. From that point forward and for the next 3 years, we as a family experienced the plateaus and steep declines. It was a roller coaster. We never knew what to expect on a given day. For my mother, his primary caregiver, this meant sleepless nights and living in a state of hypervigilance. When our physical and emotional resources were depleted to provide a safe environment for my dad, we were next forced into depleting our financial resources also. In July of 2012, unable to care for him properly at home any longer, we moved my dad to a secure dementia unit where he lived out the last year of his life. The out-of-pocket monthly expenses were daunting. That being said, I am incredibly grateful to the nurses and staff who cared for my dad when we could no longer provide a safe home for him. We could not have managed without you. In November of 2015, Susan Williams came out publicly to share her story. Reading about her experience was like reading my own. Thank you, Susan, for giving me the courage and the motivation to move past my own heartbreak and trauma in order to share my story too. Susan's story led me directly into a partnership with the American Brain Foundation and brought me here tonight with all of you. I am proud and honored to be here and to support this important work. Thank you.
Tima, wow, thank you so much for being here and for sharing what is a very personal story that obviously still has made an impact, and it's very emblematic and reflective of the importance of discovering a disease biomarker, because there are over 1 million families, as we've heard earlier, who are experiencing this type of impact on their family. It's real. So we really appreciate you being here tonight. We have disease biomarkers for many systemic diseases, from diabetes to chronic kidney disease, and for even some neurological diseases like multiple sclerosis. And, you know, disease biomarkers range in complexity and cost and invasiveness, and they can include blood tests, x-rays, other sophisticated brain imaging studies, tissue biopsies. The 2022 Cure1, Cure Many Award was created for that purpose, to find a diagnostic biomarker for Lewy body dementia. And I'd like to turn the floor over now to Dr. John Morris, who has really put in a lot of work over the past 6 years to spearhead the awards selection committee and to make sure that we were granting this major award, the first of its kind, as you've heard, to the most worthy recipients. And so, Dr. Morris, thank you for all that you've done. And please, the floor is yours.
Thank you, David, and good evening to everyone. As we've heard so movingly tonight, Lewy body dementia is poorly understood, and there are no effective therapies. As we've also heard, it is not well diagnosed. It's very difficult to develop an effective treatment strategy when you don't understand the disease or recognize the disease that is being targeted. So accurate identification of Lewy body dementia and its biological markers will move research forward into understanding the disease and ultimately its treatment. The American Brain Foundation put together an awards selection committee for the Cure1, Cure All Award. I was honored to chair that committee. We had representatives from the American Academy of Neurology, the Michael J. Fox Foundation, the Alzheimer's Association, and several world experts in Lewy body dementia. We put together a request for applications that went out in the late spring of 2021. And by the end of July 2021, we had received 21 letters of intent, people, investigators, wanting to put together a full proposal for evaluation for funding. The award committee reviewed those 21 proposals and selected 9 and invited them to put in a full application. The 9 investigative teams did that, and in November of 2021, the committee met for the final time to select the awardee. As you have heard, we found the proposal by Dr. Owen Ross to be highly meritorious and chose that for the first recipient of the Cure One, Cure All Award. Dr. Ross is Associate Professor of Neuroscience at the Mayo Clinic in Florida. He received his doctoral degree in genetics from the University of Ulster in 2003, and then came to the Mayo Clinic for a postdoctoral fellowship and has remained there ever since. He is a world expert in neurogenetics and has been instrumental in helping to characterize important genes that cause Parkinson's disease. The proposal that he has been awarded for Lewy body dementia is the development and validation of a blood-based biomarker for overt and prodromal dementia with Lewy body. He has several very innovative aims to try to work on proteins that we know are important for Parkinson's disease and underlie Lewy body dementia, but also for novel biomarkers. He is using multiple avenues to try to identify this: clinical studies, imaging studies, genomic studies, and unique ways to integrate and understand the data. He has put together a multidisciplinary and multi-institutional team, and we are very excited to see the results of this very promising research avenue. So with that, I want to give my thanks to the award selection committee, to all of the sponsors of the award, to all of the donors for the award, and I give my great congratulations to Dr. Owen Ross. Dr. Ross.
Great, thanks, Dr. Morris. A very kind introduction, and I'd like to start on behalf of the team to say a big thank you to the selection committee and to the American Brain Foundation and all its partners for this award, and really for the funding to allow us to really tackle, I think, what we've all discussed as one of perhaps the most fundamental issues with understanding Lewy body dementia, and that's really an early and accurate diagnosis, which really lays a foundation then for clinical drug trials, for understanding how the disease progresses, and for finding a way to have an intervention early in the disease, even before some of those symptoms even start to present. As mentioned by Dr. Morris, I'm trained as a neurogeneticist, and one of the things that really got me into genetics was, well, perhaps I'll blame it on the Gattaca movie, but it was that idea that, you know, once you're born, we could look at the genome and really look at the variation in each of us as an individual and determine our risk of developing Lewy body dementia, Alzheimer's disease, these age-related neurodegenerative conditions. And I think as genetics transformed, the technology is so rapidly advanced, we thought, oh, whole genome sequence will get us there, and we'll be able to look at each patient, describe their genome and say, okay, this is the disease you have and this is the disease you're going to get. But I think it became clear over the last 3, 4, 5 years with each genetic advance that genetics alone won't solve this problem, and that we're not quite there yet. And to really get an understanding of those who are at risk of developing these diseases, to be able to accurately diagnose the disease once the signs present, we need to bring together an array of different forms of data, including, as Dr. Morris mentioned, genomics, imaging, the clinical phenotyping that's done by some of the excellent movement disorder and behavioral neurologists who see these patients on a daily basis, and bring some way to get all that information together and to tie it in with a biomarker that's measurable and is easily accessible. So I have just a couple of slides, some very basic slides to sort of highlight what the proposal's really going to try to get at. And one of the first things we did, that myself, and this initiative is really led by Dr. McLean, Dr. Bove, and myself, was to really think about what's needed. And we felt strongly that what was needed was a blood-based biomarker, a biomarker that's not going to require expensive imaging necessarily, or lumbar punches, or something that's easily accessible, and that's blood. We decided we'd go and look for the best people who are working on blood-derived biomarkers and have worked on Lewy body dementia. And Dr. Sid O'Brien has worked on this at the University of North Texas for almost 20 years, developing these blood-based, looking for different types of biomarkers within Alzheimer's and within Lewy body dementia. So we reached out to Dr. O'Brien and said, we're going to put this together, can you help us? And then Dr. Isidore at the UPenn had really developed some innovative ways of looking at biomarkers in the blood that were coming from the brain directly. So we reached out to David and said, can you help us pull this together? And then within the Mayo Clinic, one of the things we have really good access to is samples and clinical patient biospecimens and descriptions, clinical descriptions that have been collected over many, many years, imaging data, genomic data. So we reached out to our colleagues in neurology, Dr. Savaga and Dr. David Jones, who are bringing together some innovative machine learning, AI, artificial intelligence approaches to really try to bring all these different data sets together and all these little pieces of evidence, because in some ways that's really what's been sort of collected over the years by your neurologist, is each little piece of evidence that really determines how we can get to that early and accurate diagnosis. So we decided to take a team science approach to this and really come at it from different angles that we hope will then produce a blood-based biomarker that will really define the diagnosis as being Lewy body dementia. But as you'll hear, hopefully we'll go beyond, and as is the hope with Cure One, Cure Many, is that it will also help with other related disorders such as Alzheimer's and Parkinson's disease. So the team we put together is really strong. The project itself, if we can go to the next slide, the project itself is really coming at it, like I say, from the blood perspective. And Dr. O'Brien, has looked at different proteins in the plasma. So we take the blood, we spin it down, and we can look at some of just the protein levels and how they are different between patients and control healthy individuals. Then the other parts of the proposal are it's a really innovative way of looking at it. There's these little packages called extracellular vesicles, or you can just think of them as little packages that go from brain cell to brain cell. And just like the USPS, sometimes they get lost in the way and they end up in the blood. So we get these little messages that have unique signatures that reflect what's going on in the brain that are then passed through into the blood. And we can detect those in the blood. So the other part of this project is finding those packages in the blood that are coming directly from your brain cells and looking to see if the toxic species of protein that aggregates in the brain can be measured within the blood. And Dr. Isidore at UPenn has developed this little microfluidic chamber that basically you'll put the blood plasma through, and it'll actually select out those little packages that were going between brain cells, and then we can characterize that to show that what's going on in the brain we can now read directly from the blood. And that's the hope here, is that we're going to get a signature from the brain that defines Lewy body dementia and detects that protein. Some of you may have heard of it before, alpha-synuclein, that's really driving some of that aggregation within the brain of the patients, and define that from the blood. Now, once we have these 3 or 4 approaches from within the blood, we can take that back. And one of the nice things here at the Mayo Clinic is that there's this wealth of clinical data and imaging, and we have whole genome sequencing on all these patients. But now we've got so much data, of course, how do we actually analyze it? How do we define that signature profile that we're going to see in a patient versus a non-affected sort of healthy individual? And that's where Dave Jones and his team and Dr. Isidore and Dr. O'Brien are all using machine learning approaches where we can take these huge, vast quantities of data and really simplify it down to a set signature that will sort of be the telltale sign that we're seeing this Lewy body dementia profile within the patient. And the hope is, of course, that we can do this early in the disease, perhaps even before the clinical signs are presenting. So I sometimes like to think of it in the sense that, you know, my family history is all heart disease. So my cholesterol was measured. And as soon as the cholesterol started to go high, they said, go exercise and eat healthy. And of course, we're going to do the same thing. And I'm sure Dr. Bove will totally agree, eat healthy and go exercise is the first thing we're going to say. But if when you have your general blood test done on your annual clinic visit or whatever, we have these biomarker assays that can quickly be read. As soon as there's any sign of anything, perhaps we can get some interventional strategies. And I think one of the things that we're really struggling with in neurodegenerative disorders, Parkinson's, Alzheimer's, Lewy body dementia, is, you know, the continued sort of lack of exciting results from clinical trials. And one of the concerns is, of course, that we're starting too late. So hopefully this would give us very early, accurate biomarker signs. And that's really the goals of this proposal. So if you can go to the next slide, really what we're trying to get at is to identify this simple blood test that could be done across the board and even in the healthy population to screen for those at risk or those really early development, but to also define the diagnosis for those that have started to present symptoms. And to make it accessible and easily accessible across different labs and not be super expensive where only certain things can be, certain groups can afford it. And then the nice thing is if we can detect these signatures or even the alpha-synuclein protein signature early in the blood, it'll really allow us to say for definitive diagnosis that this is a synuclein-driven disease. It'll also allow us to identify those at the very, very early stages of disease, but it'll also let us know that in, say, Alzheimer's disease, where perhaps 20–30%, maybe more, have coexisting synuclein pathology, it'll really inform into those patients who could benefit from a synuclein therapy. So the assays may directly have a benefit within Alzheimer's and within Parkinson's, but also the approaches we're using, looking at the small packages that are passing between brain cells, the microfluidic chamber to isolate those small packages in the blood coming from the brain could indeed in itself provide a technology that could be then used for Alzheimer's disease or for Parkinson's disease or for any of these brain disorders. Collecting that signature from the brain, but getting it from the blood could really move the field forward. I'll stop there.
Well, Dr. Ross, that was a fantastic presentation. Thank you, and congratulations on behalf of the American Brain Foundation and all of our partners on this very prestigious and award. And so my hats off to you and your colleagues. We can't wait to see what unfolds from this research, because as you said, this has implications for profound implications for Lewy body dementia, but also for other similar brain diseases like Alzheimer's and Parkinson's disease. So thank you again for that presentation and congratulations. And speaking of these other brain diseases and the implications that your research is going to have, we're really honored to partner with the Alzheimer's Association and with the Michael J. Fox Foundation for this initiative. So we're very pleased to actually have Dr. Heather Snyder here with us tonight, who is the Vice President of Medical and Scientific Relations at the Alzheimer's Association. So Dr. Snyder, thank you for being here. Please join us, give us a few words if you would.
Absolutely, and thanks so much for having us. The Alzheimer's Association is committed to funding research into the causes, the treatment, prevention of Alzheimer's, and all dementia, including Lewy body dementia. And we're so proud to be partnering with the American Brain Foundation, the American Academy of Neurology, Michael J. Fox Foundation for Parkinson's Research, the Mary E. Groff Charitable Foundation, and so many others that made this program and award possible. As I think we've been hearing and we've been talking about, Lewy body dementia is a complex disease, and it's really by working together as partners that we're joining all of these organizations, these groups, Dr. Ross, the scientific advisors, and the larger team, and then it's working together that we will move forward to advance this important work. And it's really clear that in order to do this, our power and our impact together is greater. Work like what Dr. Ross and the team will be doing is going to help not only lead to a more accurate diagnosis and new tools and new research, but it will also help people living with the disease get specific care and support that they need. And like the name of this important initiative, by learning about and developing the tools for Lewy body dementia, this will advance the entire dementia research enterprise. We join all of you in congratulating Dr. Ross and the team. And again, thank you to the American Brain Foundation and their team and all of the partners that are working with us to move this work forward. Like all of you, we really look forward to seeing the outcomes of this project in the very near future. Thank you so much.
Thank you so much, Dr. Snyder. Thank you for your partnership and leadership. And now we'll hear from the Vice President of Research Programs at the Michael J. Fox Foundation, Dr. Jamie Eberling. Dr. Eberling, thank you so much for your partnership and for being here tonight.
Thanks so much. First of all, I want to thank the American Brain Foundation, the Alzheimer's Association, and the American Academy of Neurology for their partnership in this program and event. Thank you too to Susan Schneider-Williams, who has been a friend to our foundation. The Michael J. Fox Foundation's mission to cure disease and develop new treatments relies on the power of science and the power of community. This program does the same. We're proud to contribute to the Cure One, Cure Many Award to serve a broad patient community with the most cutting-edge science. Parkinson's disease dementia is a Lewy body dementia. We hear often from our patient population and their families the great impact of this condition. Better tools, especially those with wider application potential, such as a blood test for LBD, would be game-changing. Diagnosing and even predicting this dementia would allow patients and families more answers and guidance to plan their lives and care. And one day that care could include a treatment to prevent LBD. The outcomes of Dr. Ross's project may speed those therapies as well by measuring the impact of intervention. On behalf of the Michael J. Fox Foundation and the Parkinson's community, I want to again say how glad we are to be part of this program and how enthused we are by your work, Dr. Ross. We will cure many together. Thank you.
Thank you so much, Dr. Eberling. And thank you all for the support on this really important initiative. It's truly amazing work, and I'm excited, we're all excited, and many families are waiting to see where this goes. So now I'd like to open the floor actually to questions about the Cure One, Cure Many Award and the research that's planned. We're fortunate to have members of Dr. Ross's team here with us this evening, Dr. McLean, Dr. Beauvais. And so please, many of you have already been adding your questions to the chat feature. Please continue to do so. And also, if you would like to actually ask your question live, just raise your hand and we will open your mic and have you answer the question in person. But while we're waiting, maybe I will ask our esteemed investigators here tonight. There are a number of questions that have come through, and I'm coalescing or bringing together a number of them that asked about distinguishing, at least currently, in the absence of a disease biomarker, between Lewy body dementia, Alzheimer's disease, Parkinson's disease with dementia. How do you as clinicians now approach this diagnostically, and does brain imaging help you, PET imaging? Maybe you could speak to the audience about this.
I'm going to throw this one to you, Dr. Beauvais.
Thanks, Owen. Thanks, Dave. Brad Beauvais here. Excellent question, Dave. For clinicians, we rely on a constellation of certain symptoms, findings on exam, and then a number of the scans, MRI scan, DAT scans, PET scans. The issues are many of these scans are not diagnostic. They can be normal, especially early in the disease. They can also be very expensive, and many people just don't have access to one or more of these. And so a blood-based biomarker is critical. Plus, a blood-based biomarker could be advantageous regardless of race, gender, or socioeconomic status. So it just underscores the need for something that is hopefully easily accessible and hopefully relatively inexpensive, a blood-based marker.
Thank you, Dr. Bouvet. There's a question here about the currently commercially available SynTap CSF test for alpha-synuclein. What is the role of that currently? And how will this complement that particular test?
I'm happy to take that too. Pam or Owen, if you want to chime in, but I'm happy to. These are exciting advances in the field. Definitely the CSF test and the skin biopsy test are major advances. And they are already changing practice. They've only become active in clinical practice over the past year or so. For the spinal tap, though, there are many individuals who either can't or for various reasons aren't willing to have a spinal tap. Plus, they're not 100% sensitive or specific. They're very good, but they're not 100%. The skin biopsy also is very good, but some centers don't have access to that skin biopsy technique yet. Again, underscoring the desire for a blood-based biomarker.
Yep.
And I think following on, Brad, from the skin biopsy, I mean, one of the things that's a component of our proposal was recently published where they use skin in the seed aggregation assays, where you can take a normal protein and then you can add the patient sample, the biospecimen, and then you shake it up or you heat it, and you'll get this sort of exponential growth or you'll get the aggregation of the normal protein, and you can measure that. And of course, if the toxic protein, so to speak, isn't there, then you get no amplification. So if it's a synuclein-based disorder like Lewy body dementia, you'll get a signal. If it's an Alzheimer's patient without synuclein, you'll not get a signal. And that's a really nice new robust measure. And that's one of our aims, is to get that to work from the blood, from the plasma base. So basically isolating those little packages of information that's going from brain cell to brain cell. Like I say, some of them just get lost, as happens our packages in the post. Some of our Amazon deliveries just disappear. Some of those little packages just float off into the blood. And if we can capture them, that's the brain signature from the blood. And then we can see if we get that signal or not. And that should tell us whether that toxic synuclein species is in the brain or not. So we should be able to tell using that if there's synuclein aggregation, if it's a Lewy body type of disorder. And this again also works for Parkinson's perhaps. And we're trying to work in multiple system atrophy and other synucleinopathy that the Michael J. Fox Foundation heavily supports. We're trying to see, can we get this to work? So it'll be also defining in Parkinson's, MSA, and Lewy body dementia, but also in Alzheimer's, where there's co-committing sort of synuclein pathology as well. So I think that's a really exciting technique. That's one of our aims and something my lab's been working on.
Yeah, thank you both for that. There is a question here, maybe this is in your wheelhouse, Dr. Ross, for sure, regarding genetics. And you'll have to translate this for everyone once I ask this question, but is there any research being done for Lewy body dementia into the role of the brain's extracellular matrix and the role of CSPGs that make up the perineural nets in the brain's extracellular matrix? So can you translate that for us and then answer it?
Yes and no, in the sense that I don't think there's any genetics in that question, unfortunately, really. Actually, Dr. McLean would be the perfect one to perhaps jump in here as well, because I think the CSPGs and some of that is to do with sort of the movement of synuclein and the protein uptake. And how the synuclein. So one of the things we think about is how the toxic species that we're looking for in the blood moves from cell to cell and spreads throughout the brain and causes some of that spreading, and that can be taken up by different receptors and different proteins driving that. And is there ways to block that and ways to work on that? The extracellular matrix, I don't know, Pam, if you want to jump in here and help.
I'm a little, I'm afraid I can't. But the CSPGs, so it's been shown that HSPGs, which are heparan sulfate proteoglycans, are thought to be involved in the uptake of alpha-synuclein into neurons throughout the brain, and may contribute to the cell-to-cell transmission of alpha-synuclein throughout the brain, which we think contributes to the progression of these diseases because the pathology of alpha-synuclein can spread over time.
Great. Well, this one is for sure in your wheelhouse, Dr. Ross. And it says sometimes these, notwithstanding your earlier comments about genetics and the how complicated this disease is, it says sometimes these disorders run in families. What can you say about the genetics of Lewy body dementia?
Yeah, no, there's definitely, and I mean, genetics, we've got a much better understanding of genetics. And the first thing I'll say is we have to really remember that, when we talk about genetics, a lot of time we talk about a gene that causes disease and a gene that's driving disease. And there's just as many genes that prevent disease and are protective in disease. So I want to just give a shout out for genetics as it's got protection and risk. In families, what I describe in a way is that the genetic mutation in a family that co-segregates is almost like a sledgehammer type mutation. And what we see then in sporadic patients is much smaller effects. The genetics that segregate in disease, we see it in about 10 to 20% of Parkinson's disease patients, somewhere in that range that there's this family history of Parkinson's disease. Lewy body dementia doesn't seem to have quite the same familial component. We don't see large families with multiple members with Lewy body dementia. What we do see, and this is something I've talked to Dr. Bove about quite a lot, is a family history of Alzheimer's or a family history of Parkinson's within families where there's also Lewy body dementia. So there seems to be a lot of overlap. And one of the things we're seeing from the genetics of Lewy body dementia is that overlap. So if you take the most recent big study we published last year with a large consortium effort, and this was also led by Sonia Scholz up at NIH, the whole genome sequencing effort in Lewy body dementia, the top hit, or one of the biggest risk factors for Lewy body dementia was ApoE, which is a major Alzheimer's disease risk factor. The second hit was alpha-synuclein, which is, of course, the largest PD risk factor. So we were seeing this combination of genetics of Alzheimer's and Parkinson's coming together. And it seems to be more at that sort of level of population risk rather than families with large segregation. That's not to say there isn't families that have Lewy body dementia that's caused by a point mutation in the alpha-synuclein gene or extra copies of the alpha-synuclein gene. That was one of the really interesting findings, almost 20 years ago now, was that in some of these families that have really early onset disease and very aggressive disease, we all as a normal population have 2 copies of the gene, and they had 4 copies. And they get a really severe early onset form of Lewy body dementia that's segregated in the family. So there is rare familial forms out there. They do inform into the sporadic disease, but there seems to be less of a pure Lewy body dementia phenotype segregating than perhaps we've seen for Alzheimer's or for Parkinson's.
Thank you. There's a couple of people with their hands up to ask questions. Kita Delet, I hope I'm pronouncing that right. Do you have a question?
Yes, this is Mike Goldberg. Can you hear me?
Yes, we can.
Thank you so much. This has been really exciting to hear this. And I am a person who is living with dementia. I'm an international dementia advocate. And I have to tell you, I actually commend you to see that people are getting together to address multiple neurological conditions, because I think that's the key. I think more of us need to partner together, whether it's Alzheimer's, Lewy body, or other diseases. But I think that's the only way we're going to make headway, because some of these diseases are so small, they're almost kind of considered rare diseases. But together as a group and partnering together, I think we can make so much more of a difference. So I commend you guys for taking a first step to do something.
Well, Mr. Ellenbogen, thank you for that. We couldn't possibly agree with you more. I don't know if our award winners would like to make a comment.
No, I totally agree. And I mean, I think what we're seeing is there is a lot of overlap in these disorders, and certainly by understanding how one of them works, will inform into how the others work. And you know, what we do for one, I think really does feed into many. And I think that's where the one cure, you know, cure one, cure many really does play a role in that.
Yeah, it's been our guiding principle for a long time, and for precisely the reason, Mr. Ellenbogen, you just articulated. Thank you. I'm not sure if Kita Delet has a hand up as well. Or maybe that was Mr. Ellenbogen. So there's another question here. How can the Lewy body dementia community work with drug sponsors to build in and fund expanded access programs into their LBD disease modifying trials from the beginning stages of their trial design planning, so that LBD patients who want to try a medication can access it?
I'm happy to chime in here.
Please.
That's an excellent question. First, we need therapies that attack part of the critical cascade of events that are related to Lewy body dementia. So alpha-synuclein, inflammation, and we know over half of people with Lewy body dementia have some degree of Alzheimer's changes too. And so first we have to know what targets to hit. We have to know what companies are working on which agents in any of those targets, and then try to work together, which is not an easy thing, working with multiple companies. Pursuing compassionate use is very difficult, and we probably don't have enough time to discuss that in detail. But first, we need targets that have at least some degree of safety and efficacy before we can even approach compassionate use.
Yeah, thanks, Dr. LeVay. There is a couple of questions here. The first one is, are there any characteristics that you can identify that make a person more susceptible to, or you can identify a person at risk for Lewy body dementia before they actually start manifesting the telltale symptoms or signs? And then the second question is, and this has been asked a number of different ways, do Lewy bodies, which are found in Parkinson's disease and in Lewy body dementia, do people with Lewy body disease or Lewy body dementia always eventually develop Parkinson's disease? So maybe you could clarify those two questions.
Dr. Bove, do you want to start with some of the sort of preclinical signs that I think really do define, which I think are a real insight into the disease process, and again give us opportunities to think about an early diagnosis?
Yes, there's a lot of excitement in the prodromal and preclinical aspects of Lewy body dementia, as well as Parkinson's disease. One of the strongest risk factors is a particular sleep disorder known as REM sleep behavior disorder. Normally, when we're in REM sleep, which is the dream stage of sleep, we're paralyzed and we don't talk much, we don't yell, we don't flail our limbs. But in that particular sleep disorder, that normal paralysis is lost, and people can yell, scream, shout, kick, and injuries can happen to themselves or to their bed partner. And this is pretty common to precede the onset of Lewy body dementia or Parkinson's disease by years, if not decades. And so there's a lot of work now trying to study people with REM sleep behavior disorder, and this may be a key window of intervention. And this particular effort that we've been awarded, again, very thankful for this, we'll be looking at the biomarkers in those with REM sleep behavior disorder if they do pan out in overt Lewy body dementia. Other things are more nonspecific: depression, constipation, loss of smell. Now with COVID, you don't know how to make sense of loss of smell. So RBD is pretty sensitive and specific as a prodromal feature, but there are many other things that may be a risk factor clinically for somebody who develops Lewy body dementia later in life. Anxiety is another one, mid to late life anxiety.
And there's also some genetic factors. So if you are ApoE4 positive, that can increase your risk of Lewy body dementia. It also is observed in Alzheimer's, so it doesn't differentiate, but it definitely increases the risk of Lewy body dementia. And then there's the genetic factors that increase your risk of Parkinson's disease. I think maybe you've heard about GBA mutations and how being heterozygous for GBA mutations. So if you're ApoE4 and you're heterozygous for GBA mutations, which are both relatively common in the population, that then makes you much more likely to produce a Lewy body dementia phenotype than the head down the amyloid or Alzheimer's disease phenotype. So if you tie in some of those genetic factors and then bring in some of the clinical signs, like do they have RBD and are they E4 positive and have they GBA, then you start to move along the sort of path of evidence towards saying that's Lewy body dementia. And then I think if some of the assays we're developing also then feed into this, and we can bring all that information and with Dr. Jones and Isidore and O'Brien and use machine learning to really pull together all the information in the one model, then we'll be able to get a really accurate diagnosis for Lewy body dementia.
Yeah. Very good. Thank you for that. Maybe the last question, there seems to be a number of people asking about Lewy bodies and why. I guess the question here is, if people with Lewy body dementia, do they develop Parkinson's disease or do they develop something that looks like Parkinson's disease? And what important implications does that have regarding treatment? So from a clinical standpoint, what is the difference between the Parkinsonism that patients with LBD have versus Parkinson's disease without LBD?
I'd be happy to elaborate on that. It's an excellent question. And Lewy body disease and Parkinson's disease are very complicated disorders. This alpha-synuclein protein, which we believe is a driving force behind the illness, it can affect the nerves in the body, outside of the brain and spinal cord, in the spinal cord itself, and in various parts of the brain. When it affects the base of the brain that's involved with the dopamine system and other motor circuits, Parkinson's disease is the clinical manifestation. But in some people, it skips or doesn't affect that part of the brain that much early in the course. And there's more of a thinking or behavioral change before Parkinsonism evolves. And in some people, they never develop overt Parkinsonism, even later in the course of the illness. So this really is a complicated disorder. Some of this is also a little bit of semantics. Parkinson's disease, Lewy body disease. Lewy body disease, we tend to refer to it as the histopathologic disease. Parkinson's disease is more the clinical phenotype, but some people also equate it with the pathologic disease. So even the nomenclature is a bit confusing, to be honest.
Yeah.
And the species, I'll just add, the species of the protein synuclein can be different. So it can come in different flavors as well. And that's why we're really lucky in some ways, because we have Dr. McLean, who is an expert in the synuclein protein as part of, and really put this proposal together as well to sort of really get an understanding of the different types of this protein. And Dr. McLean's been working on this for 2 decades, would you say, Pam? And it knows everything there is to know about the synuclein protein. So that's really, really going to help.
Well, unfortunately, Owen, I think if we knew everything we needed to know about alpha-synuclein, we wouldn't be sitting here tonight. But that's the goal, anyway.
Well, I said I've asked the last question, but this is all 4 patients and 4 families who are impacted by Lewy body dementia. So I'll end with this question. Beyond clinicaltrials.gov, is there an LBD focus group that currently compiles a current and up-to-date list of enrolling and/or upcoming LBD clinical trials for families to refer to?
The best source is clinicaltrials.gov. There are a number of groups that are actively engaged with the pharmaceutical industry and clinical groups, but that is the best resource, realizing it is a kind of a clunky website to use, but it is the best source.
Great. Well, look, thank you all very much.
Michael J. Fox has the Fox Trial Finder still, I think, for related Lewy body disorder stuff as well. So I'll give a shout out for Jimmy.
Thanks,
Thanks, Owen. Thank you for that. Yeah, Michael J. Fox, it's Fox Trial Finder.
Terrific. Thank you. And thank you all for joining us tonight. This was a very important event for all of us. for obviously for the investigators who were given this prestigious award, but also for the American Brain Foundation, the American Academy of Neurology, and our partners here, the Alzheimer's Association and the Michael J. Fox Foundation. So we thank you all for being here tonight. We thank you for joining us, and please feel free to peruse our upcoming webinars in our resource library. You've made this evening one to remember, and we'd like to take a moment before signing off to give you an opportunity to donate to this Lewy Body Dementia Fund. This research will undoubtedly not end after this 3 years, and we'd like to continue to support research in Lewy body dementia. So please, every donation gets us closer to a world without brain disease and gets us closer to a diagnostic marker for Lewy body dementia, and ultimately to treatments and eventually the cure for this disease. So thank you all for being here tonight. Congratulations again, Dr. Ross, Dr. Beauvais, Dr. McLean, and to the rest of your team. Everybody have a great night. Stay safe.